Thank you very much, and I'd like to thank Gorm Breeson and the committee for inviting me. It's my pleasure to be here. So my background is in biology, and in particular I'm interested in cellular and molecular mechanisms that are associated with longevity in animal species, primarily mammals. We work with some birds, and we're really interested in what makes animals live longer. Some species live longer, and sometimes we'll do that within a species, so just looking at mice, for example. And it does turn out that a lot of that has to do with time management. And there's all kinds of ways that time comes into play here, and I'll just pick out a few of them today that I've chosen based on what I think you're interested in. I hope I'm right. So I was a bit worried when I came that no one would know of my small university in Canada, but it turns out to be quite famous here in Portugal, and I've seen nothing but Superbrock since my arrival. So there's a few scales of time from very short to much longer that are relevant here, and I'm going to pick out just a couple of them to talk about today in 25 minutes. But we can think about time in terms of biochemical reactions, very short time frames, and I call that metabolic time. So this is my favorite enzyme, superoxide dismutase. We do a lot of work with this. I have a background in mitochondrial bioenergetics, which you'll see a little bit of in a second. And so we're very interested in reactive oxygen species handling, though I won't talk about that today. So metabolic time. Time is important in terms of diurnal time scales, so daily changes in things like this is showing human temperature, core temperature throughout the day, and how it is actually following a very set pattern that is time-driven. In Canada, maybe not so much here, or we're in your own country, but in Canada we're very concerned with seasonal time changes, and in particular things like hibernation. Animals that hibernate well are able to control time in a biological sense. They're able to slow things down, and I don't mean metabolism so much as actually growth and developmental processes seem to be more important. This is one I'm very interested in, and we've been spending a lot of time putting together a database that's now available online where we're looking at lifetime or maximal lifespans of different organisms, including humans, and all of that data is being compiled and made available to people to study and look for trends and look for a deeper understanding of what's going on. And then finally, of course, in biology, we're often concerned with evolutionary time. But today, I'll focus my attention and yours on metabolic time and growth and development. Growth, development, and lifespan. I think of it mainly in terms of lifespan, but you'll see how those things are quite connected. This is a schematic showing cellular energy metabolism. And it's a good starting point for this talk. I realize it's elementary, but it's a good reminder. We often learn about cellular biochemistry and metabolic pathways in a very complex way, but actually because they can be broken down into something much more simple, that having that level of understanding helps us to understand what's going on, and I'm going to relate this to experimental design, how to think about differences between species, because some of you are using animal models to understand what happens in people. So what we have is fuels that come in, shown on the left here, enter these various biochemical pathways, many of which are in mitochondria, the citric acid cycle, electron transport chain. The oxidation of those fuels builds up the membrane potential, and that potential is used to drive oxidative phosphorylation. That is the process of oxidative phosphorylation. It's the phosphorylation event, ADP being phosphorylated to ATP, that's the critical thing there. Why do you make ATP? For the sole purpose of actually having it used up. ATP hydrolysis back to ADP fuels all kinds of processes. It fuels pumps of ions, it fuels myosin ATPases for contraction of heart and muscles, it fuels protein synthesis. The way metabolism in cells is actually organized is around this central molecule. And we can think of ATP turnover, and this is going to be critical to what I talk about next. ATP turnover is simply that rate of ATP production to its use. And a really critical concept is just that ATP, we don't store it. It's not stored in cells, it's continually cycling. And it's synthesized, it must be synthesized at a rate that matches its use through hydrolysis. And so what we see is that cells that have a high work rate actually have high ATP turnover rates. And because ATP is being produced primarily in mitochondria, they tend to have lots of mitochondria. And because mitochondria, to do oxidative phosphorylation, need oxygen, that terminal step turns oxygen into water, that means that they're also consuming a lot of oxygen. This is going to be relevant in terms of ischemia reperfusion. So if we think about the hypoxia tolerance of human tissues, of any human tissue, we see that it's quite variable. And I know some people work, I've seen lots of posters on brain ischemia, and tissues like brain tissue, heart tissue, very susceptible, very sensitive to interruptions in oxygen supply. And the reason is they cycle ATP so rapidly that as soon as you interrupt the production of ATP and you've done nothing to change its usage, then the cell just uses up all the available ATP very quickly and you're in crisis in a very short period of time. On the other hand, other types of cells, skeletal muscle is an example, but also really slow metabolizing cells, skin, cartilage, bone, tend to be quite tolerant to hypoxia, and part of that is just about time. How much time does it take to deplete the 3 millimolar ATP level that's in a cell? If you're actually cycling that very slowly, well then the rate of depletion when you interrupt the production side is also quite slow. And that's what I'm showing here, and I use this graph, this schematic often when I'm talking about this topic. The work here can be anything. So that's the myosin ATPases, those are the sodium potassium ATPases, all those things that you remember learning about a long time ago probably. That is what's going on inside of all these cells, and we see the cumulative total of that in terms of what's going on in a whole body. And it's got to be balanced with oxidative phosphorylation, and you don't have oxphos without oxygen. Why is all that important? It's important because not only do you have these differences within a species, within people, in terms of sensitivity of brain versus skin, for example, to a disruption in oxygen supply, hypoxia or anoxia, you see these differences between species. And because in science, given that mice are small and rats are small and they're cheap to cage and keep in a facility, and we have knockout mice available, those are the models that we're using often for people. I certainly saw some good piglet work as well, but we see a lot of mouse and rat work. They're small. And when you think about small animals, you have to think about metabolic rate allometry and the fact that the per cell rate, metabolic rate, is so different in a small animal or a small species than in a large one. So I'll give you this comparison to illustrate that. Isometric relationships are those that hold 1-to-1. So as you change one value, body mass, you get a proportional change and an equal change in the other value. An allometric relationship is one in which you get a smaller change in the metabolic rate, for example, as you increase body mass. So you can increase body mass by 1,000 times, but you won't increase, as you get to bigger and bigger species moving from a mouse to a human, increase body mass 1,000 or 2,000 times, but you won't get that same increase in metabolic rate. There is an economy of scale, is the easy way to think about it. So we can do the math. A 25-gram mouse compared to a 50,000 gram or 50 kilogram human. It's about 2,000 mice per human. But if you added up all of the individual metabolic rates at rest for the mouse and compared it to the equivalent mass of human, what you would find is that those 2,000 mice consumed oxygen had a metabolic rate 10 times greater. Why does that matter? Okay, so it matters because this is happening at a cellular level as well. So those are measurements I was showing previously, just whole body oxygen consumption. But this manifests at the level of the whole body because of changes in individual cells or differences in individual cells. And this is shown well for hepatocytes, but it's true, and that's just because of experimental convenience. They're easy to isolate compared to some other cell types. But this is true for many, if not all, cells when we compare across species of quite different mass. And what's shown here on the y-axis is the rate at which those liver cells, hepatocytes, consume oxygen versus the body mass of the animal they came from. And this is old data, but it's useful to bring up. This comes from the lab of my postdoctoral advisor, actually. And what they showed was that when in these freshly isolated hepatocytes, just fresh from the animal, quite different rates of oxygen consumption. So the largest come from horses, the smallest from mice, and over that breadth, that range, there's about a 10 times difference in just the resting rate of oxygen consumption. Why does that matter? Well, here's the rat and pig. There's no human in this study. No humans were willing to give bits of their liver for this. Understandable. But a pig of 37, so 37 kilograms, quite a small person. So this is going to be an even bigger difference. But the rat to human is at least a more than 2 times difference in oxygen consumption rate in these tissues. And then if you look at an experiment that is a typical experiment, I would say, done, this time in hepatocytes, but often in other types of tissues, someone came along and exposed these human cells, and in this case, rat cells, hepatocytes, to anoxia, a period of anoxia. And they chose to, when they're designing their experiment, they chose 2.5 hours. And then they introduced a period of reoxygenation. That's all fairly typical. But what do you think that means, 2.5 hours to the slowly chugging along human hepatocyte versus the quickly metabolizing rat hepatocyte? The latter is going to deplete its ATP much more rapidly, and that's when trouble starts. That's when xanthine oxidase begins to produce radical species of oxygen, and when you have all kinds of secondary injury that occurs. And of course, you see it upon reoxygenation, but it's occurred during that anoxia phase. So I would argue that that needs to be thought out differently. You need some awareness of really what you're modeling is how long does it take to deplete the ATP and run into problems. And if it's being depleted at different rates, then those are going to be different answers for different species. Okay, so I'm showing that there. So, a faster cellular metabolism in smaller species, I'm saying, is an important consideration experimentally. I'm not sure how you solve the problem, other than you need to know for your individual species and the tissue you're interested in, you need some background information. A lot of people use cells in culture to do their work, which is great, and I do tons of this myself. We have a huge collection of fibroblasts and myoblasts, muscle cells, from many dozens of different species of mammal. And those are good, powerful models, but there's a couple things that I need to mention about how that would look in terms of doing those experiments. One thing that we were interested in my lab was whether whether metabolic rate allometry manifested in culture when you have cells growing in a dish. We didn't know if there was some intrinsic genetic component that told a mouse cell to metabolize more quickly, or if it was just because that mouse cell happened to be in a mouse, and the mouse required that kind of speed of the cell, and the cell responded. There are very well-characterized pathways for upregulating mitochondrial abundance. So we went out and we collected all these myoblasts from we made primary myoblast lines from all these different animal species. And we chose myoblasts because we can proliferate them in culture, get lots of cells, and we can differentiate them into something that looks very much like a myofiber, a multinucleated, striated cell type that's capable of contraction. So we did that experiment with about 10 different species, and then we did a whole lot of work, and I'm just going to show this one summary graph here, a whole lot of work to characterize their metabolic profile, I suppose, in culture. And what we found is, in fact, there was no allometric scaling. There was no allometric scaling. They all adopted a very similar metabolic rate, which tells us that it's the demands of being in a particular organism that's making this change. But it also tells us, from an experimental point of view, that if we're looking at cells in culture, maybe we don't have to think about those differences between species in metabolic rate because they don't exist in culture. And then I drew this picture to illustrate the other thing that I discovered at that time, which is that what happens to those myotubes, those myofiber-like cells, when they're in culture, is that they don't adopt a really fast metabolic rate. They adopt a very, very slow metabolic rate that's quite similar to what you find if you take a rat and you denervate it. If you cut the motor neuron serving a particular area of muscle, then it will go on vacation. It will stop working. And if you look at the levels of mitochondrial and other metabolic enzymes in that denervated muscle of rat, it's very similar to what we measure in culture. So in other words, in culture, the interspecies differences are gone, and everything's just slowed down. Those are both important considerations. Again, if you're thinking of an ischemia reperfusion type of model, it's important to understand how resistant cells in culture can be to that, simply because of their relaxed metabolic rate. So these are the two concluding thoughts that I thought might be relevant to this audience. Small mammalian species like rodents cycle intracellular ATP quite rapidly, which is an important consideration for ischemia reperfusion types of experiments. The difference is absent, though, in cultured cells which have this low metabolic rate, and again, something to consider. The second area, so switching gears now, the second area I said I would speak about is this idea of lifetime. So growth and development and lifespan, and we normally think of maximum lifespan in our own work, so the maximum lifespan that any individual of a given species will achieve. Sometimes we think of it as lifespan potential. And of course, you've all observed that there's this very strong correlation between lifespan, longevity of a species, and the body mass of that species, which is not to say that there are no exceptions. There clearly are things like naked mole rats and bats and birds, although this is just mammals I'm talking about here. So there are definitely exceptions, but nonetheless, there is this underlying, highly significant correlation. You're bigger, you're going to live longer. If you're bigger, you're also going to grow much more slowly. So the rates of tissue growth are actually negatively correlated with the body mass of a species, of a given species. So this is adult body mass on average of that species. And they're also highly correlated with lifespan of those species. All of these things are correlated with each other, and they can be teased apart statistically, but it's not necessary to do that right now. What's going on there? Well, those short-lived species that are actually growing and developing really quickly, and they need to. If you're a mouse and you don't live in a cage in the lab and you're going to be eaten, you need to grow as quickly as possible. There's great evolutionary pressure to grow rapidly to the point where you can mature quickly and reproduce before you're eaten. So that's what's there. What kinds of growth factors, we might ask, are actually involved in pushing that process forward so quickly? And I had that question a few years ago, and so I published this work at that time. And in this case, in that graph, every single one of those dots, or diamonds, is an individual mammalian species. So there's 36 of them. And in each case, the insulin-like growth factor 1, which is an important growth factor, and has a fairly well-characterized role in lifespan and longevity and aging as well. What we see is that if we measure the level of that hormone in blood, in plasma, of adults, from all these species, we see this negative relationship. So those bigger species are growing more slowly, and they also have lower levels of IGF-1. And my colleague, Vera Gorbanova, when I presented that work, she was actually really disappointed because she was doing the same kind of work, but just recently has published the complementary data to this, which is to look at IGF-1 receptor. It's a hormone. It's a growth factor. Without a receptor, there is no response. So she was able to go in. She takes a very similar kind of approach, and measured the IGF-1 receptor levels in species, the same kind of distribution of species, and she found something very similar. So both IGF-1 hormone levels and the levels of its receptor are negatively correlated with longevity and body mass. And those are shown, both of those correlations are shown here. So we know that these smaller, shorter-lived species generally have quite enhanced IGF-1 signaling. That's important, again, I would say experimentally. I think it's intrinsically interesting. It's also important experimentally because if you're thinking of an injury model and what you're interested in is the rate of repair of the injury, then you need to think about the fact that we only know this, by the way, for IGF-1, but I would be willing to hypothesize that you would see something very similar for other growth hormones, growth factors. And it would probably be the case that all of these would be at higher levels, and so would their receptors, and the entire signaling axis would be more active in the smallest species. That will have a repercussion in terms of how long it takes to do things like regenerate injured tissue, wound healing, etc. And it's something that has to be taken into account. Some of the ischemia reperfusion models, for example, will follow up some period after that injury where there is a regeneration occurring, and this becomes an important consideration if you're going to compare mouse or rat to human. And I'm sure it underlies sometimes why these animal studies fail to extend into the clinic. And I'll just address one more quick thing that I think might be of interest and stems from all of this work. Again, if you're working with cells in culture, you're aware of limits in terms of population doubling. So a lot of people will, if they want to study a disease in humans, the easiest and quickest thing to do is to initiate like a fibroblast cell line, for example, from a patient. So there's a few things to think about in terms of those types of cell culture models, especially if you're going to be comparing, again, a human fibroblast line to perhaps a mouse. You may have a knockout mouse, a gene knockout mouse that you want to make a comparison with. There's this idea out there that in organisms that are short-lived, their cells are also short-lived in culture. And that short-lived is not really that they're dying, it's actually that they're senescing. So the replicative lifespan of those cells in culture is shorter. If you're a mouse, you might only get 9 or so population doublings out of a primary fibroblast line. If you're a human, you're going to get 50 plus typically. And that's called the Hayflick limit. That idea was noted by someone named Hayflick, and so that given the term Hayflick limit. And a long time ago, somebody noticed that the Hayflick limit, population doubling potential, was species dependent, and they correlated that with species lifespan. And then from that came the idea that if you're a long-lived species, then your cells will last longer in culture. And the reason I bring this up is because I just wanted to make this point, which is that goes away, it seems to be completely, if you remove one variable that's typically not controlled in cell culture experiments, and that is oxygen. The vast majority of cell culture work is done under what are hyperoxic conditions. And we've done a lot of work in this area where we've gone in and measured at the cell surface, so ratcheted right up beside the cell, plasma membrane, of these cells growing on a dish, and we find that if they're growing under atmospheric oxygen, 20% or so, there's a ton of oxygen there. Physiological oxygen in your body, and it turns out in almost any mammalian species body, is around 3%. So if we just look at it in terms of percent and compare that way, atmosphere, 21. When people grow cells in incubators, they take great pains to control temperature, 37 degrees. By the way, mice are 39, so that's not typically controlled. But 37 degrees, 5% CO2, high humidity, lots of glucose, usually more glucose than would be physiological as well, or often that's the case. But people often I would say most of the time, ignore oxygen. Oxygen is causing senescence in those mouse or rat cells, and if you grow those cells, if you establish these primary cultures and grow them at 3%, they'll go on for, other things can happen. They're not all happy this way, but overall, you'll greatly extend the length of time in culture. So that's actually an oxygen-driven artifact, and I'm not sure what biological significance it has, but it's something that you need to think about. So, under physiological oxygen conditions, cells are released from this Hayflick limit. So just a few concluding thoughts. Again, to reiterate, these faster cellular metabolic rates and ATP turnover rates in mice and rats shorten the time to ATP depletion and ischemia, and that can have consequence in terms of experimental design. It's not present when cells are grown from any species under defined conditions in culture. So that previous statement comes from if you were to measure cells that had been acutely isolated from an organism and had spent all of their time living under whatever conditions were present inside of that organism. Now if you've got them in a dish and you're growing them under defined conditions which are the same across the board, you see no differences of that in terms of metabolic rate. These faster developmental rates and shorter lifespans are associated with enhanced IGF-1 signaling, probably other growth factors as well, and that is certainly going to affect healing. It's going to affect regeneration of experimentally induced trauma. And then this last point is the senescence of primary cell lines in culture. And a lot of people don't work with primary cell lines, and those cell lines are typically chosen because they don't care about oxygen levels. But primary cell lines established from tissue explants care a lot about that. Those seem to be driven primarily by hyperoxia. And that's it, and I'm happy to take questions. Thank you.